Good evening, everyone. I'm Dr. Brad Schlaggar, pediatric neurologist and president and CEO of Kennedy Krieger Institute. And it's my pleasure to welcome you to Exploring the Brain. Rare disease is a phrase you may be hearing a lot these days, especially this month, as February is recognized internationally as Rare Disease Month. But increasingly, attention to rare disease is not limited to February. For instance, CNBC, the business news channel, just last month announced their launch of CNBC Cures, a multimedia project to focus on stories about adults and children with rare diseases and the research happening to find treatments and cures. That network joins a number of other media outlets that have regularly targeted their coverage on this critically important topic. And the increased focus is understandable given the fact that there are now an estimated 10,000 known rare diseases. And while having a particular rare disease is, of course, rare, having any rare disease is common, with roughly 10% of people having a rare disease diagnosis. At the same time, very real advances are being made not only in diagnosis, but in the discovery of disease-modifying treatments. Now, the designation of February as Rare Disease Month, that goes back nearly 2 decades to February 29th, 2008, when the European Organization for Rare Diseases designated February 29th, the rarest of calendar days, to be Rare Disease Day. At Kennedy Krieger, we see Rare Disease Month as a time to shine an even brighter light on rare diseases and highlight the work our clinicians and scientists are doing to diagnose and treat those rare diseases. But for us, this topic is always top of mind. In fact, we are a global leader in diagnosing, treating, and studying rare diseases. For example, in 2021, the National Organization of Rare Disorders designated Kennedy Krieger, along with our partner Johns Hopkins University, to be a rare disease center of excellence for treatment of rare pediatric diseases. So tonight we want to give you an insider's look at the innovations and breakthroughs that are happening right now at Kennedy Krieger around rare disease. Joining me tonight are 5 of my exceptional colleagues whom I'll introduce momentarily. But first, a few housekeeping notes. Your video and audio will be muted during this webinar, so please use the Q&A feature if you have questions. The Q&A feature is likely located at the top of your Zoom window, so you can click and submit a question using that mode. And we'll try to get to all of the questions during the Q&A period at the end of the panel discussion. Also, this webinar is being recorded, but your identity won't be shown on the screen, nor will we identify who asks those questions. Now, the planned panel discussion, about 50 minutes, give or take, with a 20-minute question and answer session at the end. So let's get going. We'll meet our panel now. Each of our panelists is a neurologist or neurodevelopmental specialist and a leader in their respective field of research and clinical care. Dr. Erica Augustine is an Associate Chief Science Officer and the Director of the Clinical Trials Unit at Kennedy Krieger. She's an Associate Professor of Neurology at Johns Hopkins School of Medicine. Dr. Ali Fatimi is the Chief Medical Officer and the Director of the Moser Center for Leukodystrophies at Kennedy Krieger. He holds the Blum-Moser Endowed Chair, and he's a Professor of Neurology and Pediatrics at Johns Hopkins. Dr. Jacqueline Harris is Director of the Epigenetics Clinic at Kennedy Krieger and is an Associate Professor of Neurology, Pediatrics, and Genetic Medicine at Johns Hopkins. Dr. Doris Leong is the Director of the Center for Genetic Muscle Disorders at Kennedy Krieger and is an Associate Professor of Neurology at Johns Hopkins. And Dr. Connie Smith-Hicks is the Director of the Center for Synaptic Disorders at Kennedy Krieger, and she's an Associate Professor of Neurology at Johns Hopkins as well. So welcome to all of you, and thank you for taking part in this panel tonight. And Erica, let's start with you. Let's first establish what counts as a rare disease and how common is something that's called rare?
Like you mentioned, rare diseases are top of mind for many, and around the world, you'll see different definitions of what constitutes a rare disease. In the United States, we classify rare diseases as conditions that affect fewer than 1 person in every 200,000 people. And like you mentioned, an individual rare disease is just that, it's rare. But when you take those as many as 10,000 different rare diseases altogether, we can see that the experience of having a rare disease is incredibly common. More than 300 million people in the United States, more than 300 million people around the world are sharing that lived experience of having a rare disease.
Ali, how has the study and treatment of rare disease become such an integral part of the work we do here at the Institute?
Yeah, so a very large percentage of rare diseases affect the brain and the brain's development. And given the Institute's focus on individuals with developmental disabilities, we really have to know about all these rare diseases so we can treat our patients. And over the last couple of decades, there has been a lot of new insight in terms of understanding genetic causes of the common developmental disabilities, cerebral palsy and autism, for example. We now know that somewhere between 20 to 40%, and perhaps even more, of individuals who have either cerebral palsy or autism have it because of an underlying genetic abnormality. And so that is really the root cause of their illness. And so we really need to know about them to be able to practice here. In terms of studying them from a research perspective, we have had a long-lasting legacy going back all the way to 1976. One of your predecessors, Dr. Hugo Moser, was recruited here as an eminent neurologist from Boston who was studying at that time rare brain diseases. And when he came here, he brought a group of investigators, including his wife, Ann Moser, who's still active actually today here, studying basically lipids and chemistry in the brain. And that led to many discoveries over decades that we have been really that has put us on the forefront on identifying the causes of genetic diseases here at Kennedy Krieger.
So before we go any further, let's go around the horn, and I'm going to ask each of you to share a little bit about the diagnoses that you treat, and also what drew you to the field of child neurology or neurology in general, and doing work in this space. So let's start with Jackie.
Thanks. Yeah, so I treat and research a group of disorders that are known as epigenetic disorders. They are known as epigenetic disorders because epigenetics is everything that happens to the DNA that doesn't involve changing the DNA code. And so there are a lot of these disorders, but they all share a common thread that almost all of them have significant neurodevelopmental disability as a core feature. And when families and patient groups are polled, in fact, as the most important feature of these disorders, despite that it affects many different organ systems. And that's what actually drew me to this study, is that I am very passionate about understanding cognitive impairment and what makes people think and learn differently than other people. And this group is so entrenched in cognitive impairment. Sometimes we say it accounts for up to 40% of genetic intellectual disability, that I became very interested in treating and studying these disorders.
Thanks, Jackie. Doris, you're next.
Hi. So I think my interest really started when I was even in high school and college. I was growing up at a time where we were really starting to assign genes to their respective diseases. And this was, and it was in college when we first sequenced the whole human genome. And so I think that really kind of my career has kind of followed a lot of really major important milestones in the field of genetics. And it is all very scientific based, and yet there is still something kind of magical about it, because even though we know where we get all of our DNA from, we can still be amazed by the fact that a baby looks like their parents. And so as far as my disease, my disease specialty, I specialize in muscular dystrophy. And these are all disorders that affect skeletal muscle. So there are dozens of these, and we have not even really begun to say we have discovered all of them. These can range. Some of these diseases are actually not that uncommon. Say 1 in 8,000 people. Some of them are truly, truly rare. Have only been described in maybe 5 people. And I think that really one of the exciting things about our field is just how rapidly it's growing with the discovery of new genes and new diseases.
Thanks, Doris. I love that point about how marvelous it is that and amazing that a child can just look like their parents. I'm going to take that one with me. Connie, you're next.
I care for children with a number of rare neurodevelopmental disorders, but two conditions that represent the core of my clinical practice are Rett syndrome and SYNGAP1 related disorders. Both are neurodevelopmental disorders. They're characterized by global developmental delay, epilepsy, cognitive and behavioral challenges, as well as significant sleep disturbances. Yet there are several features that set them apart. Rett syndrome is often associated with profound physical and medical challenges. Kids will lose their hand function, they're nonverbal, they cannot walk, they may need gastrostomy tube for feeding. But SYNGAP children, on the other hand, they generally maintain their ability to walk, use their hands, they sometimes have language, but their challenges are more heavily weighted towards the behavioral dysregulation, which may manifest with aggression, making it really challenging for families. As for why I chose medicine and child neurology specifically, I think it comes down to really wanting to make a meaningful difference in people's lives. I've always been fascinated by the brain, and I'm passionate about understanding the science behind how it works and helping to improve people's function. Early in my career, someone asked, why child neurology? When there are no treatments that are available for many of these disorders. And my answer was, well, treating symptoms can profoundly improve the quality of life of our children and families. And, now treating symptoms still remains a cornerstone, even though we have the emergence of disease-modifying treatments.
Thanks, Connie. Back to you, Erica.
I care for people who have a group of conditions known as Batten diseases. And these are genetic disorders that cause problems with the lysosome. That's the part of the cell that has lots of different job functions, but overall, you can put those together as jobs that help the cell stay in balance. And cells like to be in balance. The cells of the brain especially like to be in balance. And so when there's imbalance that happens because of the genetic problem, cells either die, neurons can die, something we call neurodegeneration, or they cannot work so well. And so people who have Batten diseases have cells that aren't working so well, and it causes blindness, epilepsy, movement disorders like Parkinson's disease, dementia like Alzheimer's disease, and these all come together to cause quite a devastating set of disorders that usually start in childhood. When I learned about these conditions and really realized that I couldn't provide the type of support, treatment, and guidance that I would want to, or the way that I might be able to for some of my other patients, I was really driven to try to uncover more information about the diseases and to really focus in on what is my driving career passion is developing novel therapies. We're really very much in need of new treatments for people with Batten diseases. And so I spend a lot of my time thinking about how we make sure we're moving forward in that direction so that we can impact a future generation of people who have these diseases.
Thanks, Erica. And Ali.
I always wanted to understand how the brain works at the very molecular, basic level. How do things function? Why are we the way we are, and what goes wrong in disease? And so that's why I went into studying the brain function and then into the molecular biology, and that led me eventually to study the diseases that I've been studying basically my whole career. They're called leukodystrophies. These are diseases that affect the brain's white matter. That's the cables that connect the nerve cells with each other inside our brains. And I'm a very visual person, so it was a good field for me to enter, because you see on MRI changes in the brains of these patients that you can classify these diseases for before we knew the genetic causes. And I've been doing this now for many, many years, going from initially discovering some of these conditions to now really heavily working on building new therapies to try to treat these patients that are often devastated, depending on which form of this white matter diseases they have.
I think that's very helpful to sort of set the table for the rest of our discussion tonight. And so thank you for that, everybody. Connie, let's move to you. And if you could just walk us through the process of what happens when a patient is referred to you with a rare disease. What are the next steps that take place?
Great question. So in my experience, families who come to us with a rare disorder, they usually fall in one of two groups. Some are newly diagnosed after a long, exhaustive diagnostic journey, and others have the diagnosis for a while and are seeking new specialized care. But regardless of which group they fall in, their core questions are the same. What does it mean for my child's future? And what treatments can you offer? At the Center for Synaptic Disorders, when we see patients, we have two interdisciplinary clinics, each designed to provide truly specialized coordinated care by disease-specific experts. And so the first person who engages or meets our families is our clinic coordinator. She helps gather the records from either the neurologist or the developmental pediatrician, and that would include the genetic test results, the clinical history, and any prior evaluations. I then review those records and determine which clinic is the best fit. Before each clinic day, or nurse coordinator would reach out to the family to learn about their top priorities for the visit. And we find these steps to be incredibly valuable because they make the visit more efficient and they ensure that we address the parents' and families' concerns. During the visit itself, our team rotates from one provider to the next. We discuss the patients and align on treatment recommendations, and we're doing that in real time, which is great. Parents leave with clear directives as to what are the next steps. One of the things about our program, which I really love, is that we hold this lunch session where families get to meet each other, and the broader medical team. They learn about research opportunities, they share lived experiences. And really one of my favorite parts of the clinic day is when I see families connecting and sharing contact information and building a sense of community with others who truly understand their own journey. After our clinic, it doesn't stop there. Our nurse coordinator or clinic coordinators really support families from prior authorizations, medication refills, referrals, and our research coordinators connect with families to help support their participation in research that's ongoing, whether it's our phenotypic research or the clinical trials that we're doing in collaboration with the CTU. We have two gene therapy trials in Rett syndrome that are ongoing. So, it's really exciting to see how things come together on a clinic day.
So Doris, let's spend some time and talk about the challenges that patients and families face. I think we can speak to that in the setting of your center around genetic muscle disease, but they likely generalize to the other rare diseases that we're talking about tonight. So how would you describe those challenges?
Sure. Just to give a few examples, I think Dr. Smith Hicks alluded to one, is that it can be very difficult to get a diagnosis. I think a lot of times, especially with the muscular dystrophies, the early signs can be extremely subtle. And so sometimes, there will be sort of mild abnormalities the child grows out of it. And then there are other times where it doesn't. And so it can be extremely challenging say the pediatrician who sees the patient first to know when to really pull the trigger on a genetic to look for a genetic diagnosis. I think it's also up until fairly recently, it's been really hard to get genetic testing. This has not been something that has been accessible to many people, in part due to cost and in part due to the difficulty of knowing what to order. And this is something that is very specialized in itself. And we have a really excellent team of genetic counselors that support us and help us through that. And we could not do what we do without them. I think that another aspect that we can really struggle with is the process of not having a diagnosis. I said, it's very likely that, even though we've discovered dozens of causes of muscular dystrophy, we may not even have discovered half of them yet. And so it is possible that we can do all of the testing that is currently available to us and not come up with a definitive diagnosis. And this is difficult. This ends up being a very long process. And what we try to do is to kind of characterize the disease as best we can, to make sure that that we don't let the workup go, that we revisit it at planned intervals, and that we still provide the support symptomatically that the patients need. I think another point of difficulty is that, especially with these genetic diagnoses, the diagnosis does not necessarily stick to just one member of the family. Oftentimes we discover that there are possibly many other family members who are at risk of developing some form of this disease. And this can be an incredibly difficult subject for all families to manage. And then I think another point of difficulty that kind of goes wider out is when people actually do receive the diagnosis, they will start looking into the internet to learn more about their diagnosis, and they will be confronted with just a vast amount of information, some of which is helpful, some of which is not necessarily related to their particular situation, and some of which is just completely untrue. And I think a big part of an increasing part of my role as their physician is really helping them kind of find the good information that is really helpful for them.
So Ali, just heard challenges and there are many, but despite these challenges, we agree it's an exciting time for rare disease clinical care and research. Can you speak to why? Like, what is it that's happening now that makes it an exciting time, and what are some of the causes for that excitement?
Yeah, it's truly an exciting time because we are in the middle of a dramatic revolutionary process in terms of the technology for genomic medicine, both on the diagnostic side and on the treatment side. I can give you an example. Me just not too long ago when we were, earlier in our careers, I remember watching little babies with spinal muscular atrophy, for example, in front of our eyes, losing their muscle function and dying, and that became a treatable disorder. And
spinal muscular atrophy. Just lay it out a little bit what that is.
Well, it's a so-called motor neuron disease. It's a disease that affects often very young infants very early on in life, and they lose muscle function because the nerves that innervate them basically die. And now several years ago, there have been 2 different genetic therapies that have been developed, since we know exactly what the genetic abnormality is, and so we can basically develop therapeutics that go and specifically target that genetic abnormality. And so this is the technological innovation that has really been exciting, because now we can diagnose diseases and develop targeted therapies for them. And it's basically just a matter of time and money, one could say. It's different than, say, traveling to another galaxy. We don't have that technology today, no matter what we're going to do. But we have the technology today to treat every single genetic disease. It's just that we have to do it one by one, and it takes a lot of time and investment and regulatory efforts and so on. But in theory, all of these diseases have become now treatable, and so that's really exciting because we understand now what is happening. There's also another aspect of this is early diagnosis through what we call newborn screening. And so, yes, there is only a relatively small fraction of these 10,000 or so rare diseases that have become treatable. But those that have been treatable or have become treatable, we know that the earlier we intervene, the more we can save and the more we can slow down or stop the progression of these diseases. And so there are assays called newborn screening assays. So these are tests that are done at birth on babies that are born across the board in the whole country, for example, where you can identify up to about 80 different diseases right now. And from a single, couple drops of blood and then have targeted treatments. And so this is an ongoing effort. I'm proud to say that Kennedy Krieger has been involved in the development of some of these tests. And so again, exciting times, totally exciting times. Can't be more than that.
Connie, you want to chime in also on this question of newborn screening and some of the there's excitement around it, but also some limitations that we have to keep in mind?
Absolutely. Yeah, I agree, Ollie. Newborn screening is really critical to help identify risk. But in some disorders, such as Rett syndrome, there's a lot of talk about doing genetic testing in the newborn period. And the MECP2 gene, which is the gene that's associated with Rett syndrome, that gene is excellent, which means that you can have two girls that have the same genetic change, but they develop clinically. One may have Rett syndrome, and one may just have a little bit of learning disabilities, mild learning challenges. And so one of the things that we've been grappling in that community is what is the impact of early genetic testing in the newborn period in that population. You can imagine that might put parents at increased anxiety about their child's prognosis. I think another caveat that we're grappling with is early treatment, which could, potentiate a different disorder. So in the Rett field, we're really thinking about how can we pair predictive biomarkers with genetic testing to better identify patients who will develop Rett syndrome. So that's one of the challenges in that particular field, but it's certainly newborn screening and the ability to intervene early. I think it's really exciting.
Doris, Jackie, I'm hoping you'll comment on this too. Earlier, Connie spoke to some of the really wonderful dynamics that she witnesses in her clinic setting where parents and families exchange information and get to know each other and start this journey together around rare disease. There's really a powerful role that parents and advocacy groups play in rare disease. It may not be recognized without calling attention to it. So I'd like to hear you comment on the really powerful role that parents and rare disease advocacy groups play in advancing the work that we're doing.
Hi, I'll go first, of course. I think that there are obviously many examples, specifically in the muscular dystrophy field, and probably the best example is actually the Muscular Dystrophy Association. It's been around for decades. It has raised millions of dollars in grant funding, and those grants have directly for instance, led to the identification of dystrophin as the causative gene for Duchenne muscular dystrophy in 1986. There are grants that have led to the development of Spinraza, which is the drug Dr. Fatimi was alluding to, that is the first approved treatment for spinal muscular atrophy. And so for decades, the MDA has, which is a group of patients and advocates, have all helped really advance the careers of researchers that are still in the field today and training new generations of clinicians and scientists. I think one of the trends that I've also started to see over the years is that while the MDA has a very, very large umbrella that covers over 40 diagnoses, there are starting to be a lot of smaller advocacy groups, most of which focus on a single disease rather than, say, all of the muscle diseases. And these are groups that create communities for themselves where they find other people that can that share the same experience, that encounter the same day-to-day problems, who have the same types of troubleshooting and problem solving. And although they're not necessarily large, they can play a very important role in fundraising, in facilitating trials with industry, and lobbying for to the government on behalf of people with disabilities.
Jackie?
Yeah, I agree with every point that Doris made. And in addition to research fundraising, which she mentioned, and, community building for the patients themselves, and fostering the careers of young investigators, getting them interested in that rare disease, there's also the aspect that I, and actually most of us here on this call, are what's called clinical translational researchers. So most of our research is on human beings and tissues derived from human beings. And we can't do that without actually seeing these human beings with rare diseases. And so advocacies, groups and partnerships between academics and advocacy groups play a huge role in enabling patients to come, whether that's recruitment or raising awareness or travel funds for participants, which is something that here in the United States that the NIH doesn't fund, for example. So they are an increasingly valuable partner to everyone who does research in rare diseases.
Yeah, no question. So Erica, next one's for you. Let's talk about this. I mentioned in the introduction that we've been designated a NORD Center of Excellence. What does that mean? What makes the Institute a center of excellence, and what does that designation really mean and afford us?
So NORD, the National Organization for Rare Disorders, is the leading nonprofit focused on individuals with rare diseases, advocacy, support for innovation and care and research. And NORD set out to really highlight centers around the United States who had specialized interest and expertise in care and research for people with rare diseases, to really sort of highlight these flagship programs where there was this excellence in care, just like the models you've heard described already to this evening. And Dr. Ali Fatimi, in collaboration with Dr. Ada Hamash at Johns Hopkins, led a collaborative effort for recognition by NORD for Kennedy Krieger and Johns Hopkins together to be recognized as a center of excellence. And they certainly were recognized for their outstanding care and research for people with thousands of different rare diseases here in the area. And central to the idea of a center of excellence is outstanding expertise in care and clinical knowledge and also research. So as Dr. Harris just mentioned, all of us here tonight are researchers. And I think we probably each have a story to tell of how the research, whether it's our own or across our field, how that informed how we work with families, how we recognize disease, achieve a clear and efficient diagnosis, and how we give better guidance around what to expect, as well as developing new treatments. And so those centers of excellence, that designation is where you know you're going to see those pieces go hand in hand. And that translation from all the way from the lab all the way to people, that's something that's particularly important here at Kennedy Krieger, where we're really looking to bring the most cutting edge discovery to our patients every day.
Jackie, earlier Ali made the point that we really have the knowledge and technology to get to all of these rare diseases. It's just how do you do it? There's 10,000 of them, or approximately 10,000. So a question that I think we really need to get to is, how do breakthroughs in one rare disorder benefit our understanding of others? How does leveraging studying a particular rare disease create opportunities to understand others in greater detail?
Yeah, it's a wonderful question because despite all of the great technologic breakthroughs that we have made and been talking about, despite the, academic patient group partnerships that we discussed, and despite, all of this to places like Kennedy Krieger where rare disease is at the forefront, it is still very hard to make progress, to get that time, to get that money, to be able to find a treatment for one single rare disease. And so one way around this is that many rare diseases share mechanisms meaning the way that the rare disease occurs, so with the biology underlying it, and they may share clinical features or symptoms of interest. And when that is the case, then there are opportunities for those rare diseases to be studied together, and in fact, for one progress in one rare disease to directly benefit another. So much so that some of the groups that I work with, some of the groups that do not have as much organization or money behind them, have actually donated what small amount of money they have to a group that has a little bit more money and momentum behind it, because they know that progress in that either larger or better funded disease will directly benefit their rare disease. And so this is something very important, and something I think that we all on this call and at Kennedy Krieger think about a lot.
So I'd like to go around the horn again, if you will, and ask you each to offer something that you want the general public to know about the work you do. Maybe dispel a myth or something that's exciting. And I'm going to start with you, Erica.
Well, we've been talking about research, so I think I'll stay on that topic. And I think it is critical to understand how important research is to care. It's how we build knowledge, it's how we create new opportunities and new kinds of treatment. And research is so much more than clinical trials and work in a lab. There's a whole realm of research that falls in between those two bookends and even beyond those bookends. So if there is an interest, there is likely a way for every person to participate and engage in research in a way that feels comfortable and feels right for them. And so that is something that we all do. We have these options, and it really is something that helps to advance care and advance research. So that's that integration, that tie between day-to-day research and care is something I'd like to underscore.
Ali.
No, I fully agree with Erica. I also think that one of the things that our audience needs to understand is that when we investigate these rare individual diseases and understand what happens at the molecular biological level in these diseases, that insight that we gain in these rare conditions will not just help us to help those patients with those rare conditions, but also applies to the entire field of biology and medicine, and will help common disorders and may in fact help conditions that have nothing to do with that rare disease. So I think it's important to think the implications is not just limited to to that one particular rare condition, but the discoveries that we make there applies to the entire field of medicine.
Jackie?
Yeah, I totally agree with everything that's been said. And also, I would say I would echo that I think that the way that we make advancements in these rare disease is to really think about partnerships on a very large level, right? So not just partnerships between different academic institutions or researchers, but partnerships with patient groups, partnerships with industries, partnerships with philanthropy, partnerships just across the board, and really opening up our mind to what science means and what science means broadly, and how rare disease plays an integral role in that for the reasons that both Ali and Erica just said.
Doris?
In thinking about this question, I think, for my field, in muscular dystrophy, one thing I would like to maybe even move the needle a little bit is changing this perception of who has muscular dystrophy and what is it like. I think a lot of kind of early, where people get their early information about muscular dystrophy is that they are all children, they are all in wheelchairs, and in many ways, kind of defined by being othered as a group. I think that I think it would surprise a lot of people to know that most of the patients that I diagnose now are adults. They're, oftentimes genetic diseases aren't even on the radar when we're diagnosing adults with various conditions. But I think that that my patients are teaching graduate level English. They are serving in all branches of the United States military. They go to college. They have long careers. They have children. They have grandchildren. I think that if we look at who has muscular dystrophy, I think we will find that we have so much more in common than we do in differences.
Thank you for that, Doris. And Connie?
I would agree with everything that has been said thus far. I want to tag Doris a little bit and extend that thought, because we find that in any of the rare disorders that we have discussed where kids are dramatically impacted, that those same genetic changes, perhaps with the exception of Batten's disease, but those genetic changes can be present in adults who are mildly affected. I think a lot of times we focus our genetic testing on kids, and we haven't really tested as adults. And so we don't have a true appreciation of the clinical features of the disorder over the lifespan. And I think that that is something that we really need to do much better at. And that will help inform not just our research, but the information that we're able to share with our parents regarding prognosis of the disorder.
Excellent points. And we're going to move into the Q&A part now. And that last set of points, I think, leads very nicely into one of the questions I see. The question is, if a rare disease is not caught at birth, when is it discovered? So I think talking about how just the point that you often don't think about genetic disorders in adulthood. Well, not when we were in training, that was not largely the case, but that world has shifted. So maybe pick up on that theme. If a rare disease is not caught at birth, when is it discovered? And I think, Ali, if we could start with you to talk about the various ways that, for example, leukodystrophies manifest.
Yeah, it will really depend on the timing of when the disease will present. And for example, one of the diseases I work with, it's called adrenal leukodystrophy. It basically can present in boys either at a few months of birth as a endocrine problem, a hormonal problem, where they're presenting with what we call adrenal crisis, where their body can't make steroid hormones. or they can present with neurological problems somewhere in school age, or they can look completely fine and present in adulthood. And in fact, the oldest patient I've seen who was like symptom-free was somebody who was symptom-free until his late 60s. And so that is one of the reasons why developing newborn screening and having early diagnosis is really critical, because when you identify these individuals earlier on, you have greater opportunity to intervene before it takes on. But the truth is that many of these diseases can present at any time, or may not present until some point in adulthood. And the challenge that we have, as Doris mentioned, is that many times adults trained or physicians and providers trained in adult fields don't have the experience on rare diseases because they don't necessarily think about them. We see them in the pediatric field a lot more. And so that is one of the challenges there.
Yeah. So Erica, I'm thinking also of Batten diseases, and there's a really interesting history of how those diseases were described prior to our understanding of the genetic causes. Maybe you could speak to that a bit.
Batten diseases were described exactly 200 years ago now. So back in the 1820s, these conditions were described in a family that had 4 children that one by one were affected by what we now think of as CLN3 disease. And much like this description of a family and how they were affected 200 years ago, it is in a lot of ways how things unfold today. Diagnosis is not typically made at birth. It's when a child presents with something unusual. And in this case, the unusual is rapidly progressive vision loss, rapid progression to blindness. So for a lot of rare diseases, there may be some early features that can be fairly common, whether that might be delayed walking or need for some additional support in school. But that diagnosis, if it's not made at birth, usually comes when something deviates from what we commonly see in children. And we do have this increasing emphasis on early diagnosis so that ideally we can help mitigate or lessen or slow progression of various diseases. That's not only for rare disease. We see this conversation in Alzheimer's disease, in Parkinson's disease, and many other conditions that we think of clinically in terms of symptoms as starting in one age. But when we know that actually the pathology, the disease process, likely begins decades earlier. So again, those links between how we think about rare diseases directly correlate and tie to how we think about common diseases too.
Jackie, I think this one's, each of you can answer this question, I know, but Jackie, for you, how can a disease be genetic but not hereditary?
Yeah, so that there are lots of different ways that this can be the case. So one way is, for example, the majority of the disorders that I study are usually the kids are what we call de novo. And what that means is this is a genetic change that happened newly in the child. So even though the parents don't have this change, the child does, right? And so it wasn't inherited from a parent. Now, if that child goes on to have a child, oftentimes they could pass that down to children. Another way that this could be the case is, again, with the field of epigenetics. So on top of the DNA sequence, there are things that happen that make the DNA available or not available for making proteins. And that's kind of what DNA does. It makes proteins, and proteins carry out actions in the body. And sometimes marks that are placed on the DNA or marks that are placed on histones, which wrap the DNA, can change the way that gene or that whole group of genes is made into protein, which then leads to a genetic disorder that is not necessarily inherited.
Another excellent question is, it sounds like much work has been done to identify and diagnose different rare diseases, but what about cures? Are there any on the horizon for some of these illnesses? And so I know all of you are chomping at the bit to answer this one, but Doris, why don't you start out with that one?
Sure. I think that, well, okay. So I guess, maybe the best way to preface this is approximately half of my time is spent doing clinical trials in muscular dystrophy. And at any point, my team of coordinators and physical therapists are testing multiple different drugs that at least have the potential to be disease modifying in muscular dystrophy.
Doris, can you just make a distinction or help clarify what cure versus disease modifying, that the language that we use to speak to these effects?
Absolutely. So when we talk about cure, I think that that's probably the simpler one, is that you have a treatment that makes you not have the disease anymore. And so it's and so it basically is it's if you have pneumonia, you take your antibiotics, you don't have pneumonia, then you got cured. I think that now disease modifying means that you still have the disease, right? But the manifestations of the disease and the symptoms that you experience are different, and hopefully much improved. And so in I and so in my particular specialty of diseases, there really aren't at this point there are neither cures nor disease modifying therapies for many of them. And so currently, the majority of the drugs that we are testing in trials are intended to be disease modifying. They will not necessarily make it so that you don't have the disease anymore, because your DNA is still your DNA. However, they would change the course of the disease to either slow the progression, or stop progression, or improve the symptoms.
So others want to speak to this? I mean, one of the questions might be, are there any examples out there of cures for genetic diseases involving the nervous system or otherwise? What would it take? It would take changing the genes, right? Erica.
Changing the genes in the right cells at the right time in a continuous manner. All of those conditions would need to be met to have the potential chance at a cure. I would say when we step back and really think about the big picture of medicine, cures, that true meaning of cure in the way that Dr. Leung just described it, they are few and far between. And they tend to be when there's something extra to target. There's a bacteria or a virus that came into the body and we can attack that and try to make that go away. Or there is perhaps in the setting of oncology or cancer, there's a cell that replicated and it's extra and we can try to target that and make that go away. But in the case of many of the diseases that we're talking about problems that are inherent to our natural cells and the way they live, the way they survive, the way they function, and the way they have already given instructions for how the body should develop, how the brain should develop, and so many other things. When you look to cardiology, when you look to diseases of the liver, they are similarly challenged that those diseases that are inherent to the heart or inherent to the liver, the way we come about cures is through transplant and actually taking out one organ and replacing it with another. So there are relatively few things where we can give a medicine and again, a cure with the science that we have today, with the technology that we know. That being said, the idea of disease modification can still be incredibly impactful. And so we are seeing a growing number of these potential targets, as Dr. Fatimi spoke about, and a more rapid process of developing these kinds of therapies. And even therapies where we start to think about testing gene modification down to a single person who may have a unique circumstance that lends itself to considering whether that approach might be relevant. And paired with that, we are seeing the regulation of these kinds of trials at the overarching level. We're also seeing that evolve and try and attempts to evolve rapidly so that we can move forward more quickly.
Connie, can you speak to this question? Are there any new innovations for intellectual disability, presuming for treatment of, diagnosis of, and treatment of intellectual disability?
So the diagnosis of intellectual disability requires that there is both the presence of impaired cognitive functioning and adaptive functioning. So both need to coexist in that individual. And so one of the things that is routinely done by neuropsychologists, who are often the individuals who are making the diagnoses, instead of using what we call standard scores to make the diagnosis, they may use raw scale scores or age-specific normed scores to make that diagnosis. With regard to the treatment of intellectual disability, at this point in time, really, there is no, like Dr. Augustine and Dr. Lung have said, there are no drugs or genetic therapy or treatments that can target cognitive difficulties specifically. But there are a number of measures that are used, whether it is supportive care in the form of therapies, assistive communication to help engage that individual, or medicines such as seizure medicines to help mitigate the seizure and provide an opportunity for that child to learn. But at this point in time, we don't yet have treatments that can specifically target intellectual challenges. Will likely target broader difficulties that the individual might have, with the hope that there'll be improvement in their intellectual functioning.
I think this is a good question for Ali to take on. How long does it take to develop a treatment for a rare disease?
Well, we used to say a decade, going from the basic lab all the way through developing a testable sort of drug that then can be tested in preclinical models, typically animal models, and then into human trials, and then get the regulatory approval. There has been a lot of effort in trying to accelerate that process. And, Erica mentioned briefly, there is now also an attempt on what we call individualized therapy, developing selective therapeutics for an individual patient that's specifically for their molecular defect. And there you can try to get into that clinical trial stage of that one individual much faster. And that's sort of the hope. So essentially, the fastest we have seen in the last few years is that people have gone from identifying the diagnosis in the patient, to understanding the molecular defect, to developing a targeted therapy for that one, to testing it in models, and then going directly into that patient within a couple of years. The issue is that that's a little different than what would then become a sort of regular drug that's approved and paid by your insurance company. This is really just a very targeted for that one individual. And so there is a lot of regulatory hurdles that can be essentially bypassed, because it's just about one individual and not the entire world would have access to it. But there is also a lot of use of like artificial intelligence and machine learning tools right now to accelerate the process of this disease and therapy discovery. But still, the regulatory challenges remain. One of the big issues is understanding the natural course of the diseases is very difficult, because they're rare. And this is an evolving field. In fact, there's been just as we speak, the FDA, the Food and Drug Administration, has been working on some new guidance that's hopefully going to get implemented to accelerate that process. So I'm again optimistic that this process is going to get shorter and shorter as time goes on. But it is a long process, often in the lab, and many times the length of it depends on the availability of natural history data of the disease and availability of funding to develop these drugs, and then the regulatory hurdles that we have to overcome.
Erica, I think I have a couple questions I think that are important for the director of the clinical trials unit to take on. One of them is how many clinical trials for rare disease are happening at Kennedy Krieger? And then relatedly, if if it if it's a rare disease, how do you get enough patients together to do a study to know if a treatment is actually working? Ali spoke to the kind of N of one direction, but if you could speak to this issue of how do you actually do a full clinical trial for a rare disease?
We're really excited to continue to have a growing number of clinical trials to offer at the Institute. At any one time, there are around 75 to 80 clinical trials happening at the Institute. At any one time, at least half of those are for rare diseases. And that proportion that focus on rare diseases seems to be growing. Also, because that's where the field of therapy development is going. There's this increasing focus on rare disease. And so that's really something that's important that we see. And the Institute is really trying to position its clinician experts to be able to pursue and to participate in those trials. When you need to show and prove evidence that something is safe, that something is effective, and that it actually achieves the goal at hand, that takes a lot of people. And it also takes a lot of participants, or it can take a lot of participants. And so rare disease studies, they may be global endeavors. There may be sites across the United States. There are often sites around the world, all working together to try to achieve those answers and that information, and then sharing that back with the field and with the community to think about whether that represents a definite advance and success, or in the case when the outcomes aren't what was desired, what are the lessons learned and how do we actually improve the next iteration? So this is a very, very active area, and I'm really delighted to have the opportunity in one way or another to work with everyone on this call as it relates to clinical trials.
Jackie, this is a question for you. Can you speak to what actually is involved in genetic testing?
Sure. So genetic testing involves taking a sample of tissue from a person and actually then a laboratory. Sometimes that's a research laboratory like at Kennedy Krieger. Sometimes that's a large clinical laboratory that we send tests to, actually looking at the DNA and deciding whether or not that DNA is in the way we would expect it to be or has what we call a variant, and then we add this other word on top where we say a pathogenic variant. And when we say pathogenic, that means disease causing variant. And we can look at if there's too much or too little DNA, like there are chapters added or chapters subtracted from the book, or we can look at if there's typos that makes a difference in the way that that gene functions. And these days, many of those tests can be done. It used to be that all of those tests were done in blood. We still do a lot of testing in blood, but oftentimes we can do that testing in a cheek swab or saliva or skin, or even in certain cases, the tissue of interest, right? If there's something going on in the liver and that person's going to get a liver biopsy, we can do genetic testing on that tissue.
So, Connie, if you could take this one on, there was mention, I think it was Ali perhaps, that said that sometimes autism and other conditions are caused by a rare neurogenetic mechanism. Can you explain more about that?
Yes, that's great. So broadly, autism and neurodevelopmental disorders are very common. But when we think about the genetics behind it, the mechanisms behind it, that's when we have multiple different rare conditions that clinically manifest the same way. And that's in part because we have over 20,000 genes. Many of them are expressed in the brain, and the brain has a number of functions that it does. So these genes, they're expressed in the brain, mean they're functioning the brain. The neurons in the brain converge on what we call connections that we call synapses. They make circuits, and it's those circuits that execute the function that we're seeing. And so in many individuals with autism, they may have, and developmental disabilities, they may have, for example, FMR1 mutations, or they may have mutations in any other gene. So within the broad umbrella of neurodevelopmental disorders, we have many different genetic causes for those disorders.
Doris, I think direct this one to you. Very good question. With early diagnosis, would CRISPR gene editing be an option for certain rare diseases? And Doris, if you want to take it, and I think others could speak to it as well if they'd like. But Doris.
Yeah, sure. I think CRISPR can be used in several different ways. I think that and so for those of you who may not be as familiar with CRISPR, the idea of CRISPR is that this is a molecule that can target a very specific section of your DNA. And then if you have, if you complex CRISPR with other proteins, it can introduce very precise cuts in the sequence, and then replace that sequence with other sequence that you program for it. And so and this is kind of falls under this umbrella that we call gene editing. And so the and so I think that there has been a kind of I guess it was probably closer to 10 years ago now. There was like this huge public kind of huge publicity over the idea of CRISPR. And then it kind of faded away a little bit. And I think what it was is that people were raising all of the money but then they actually had to go and do all of the work to test this in cell models and in animal models. And so I think it's right now, I think we're at this point where all of that is kind of coming to the point where it's getting to be that it's kind of ready for clinical trials in people and not just in mice or in cells or tissues growing in cultures. And so I think one example is a disease that I see fairly frequently called facioscapulohumeral muscular dystrophy, and we are doing a trial of CRISPR in this disease. And so 95% of the people who have this type of muscular dystrophy have a deletion in the one end of chromosome 4. And the deletion itself doesn't actually, what's interesting is the deletion itself doesn't cause you to have like a missing protein, like in many muscular dystrophies. But what it does cause is that that it creates an environment that causes you to overexpress proteins that shouldn't be expressed. And so but what the CRISPR is going to be designed to do is to target that end of chromosome 4, and actually place chemical tags called methyl groups on that region, and make it. This is an epigenetic modification, not a genetic modification. But basically, kind of create this DNA modification that prevents genes from being inappropriately expressed. And so, the way that I always describe FSHD to my patients is that for some reason there is a problem in your security system. And so, when the guards change or something like that, then there is an opportunity for the prisoners to escape and wreak havoc in society. And what the CRISPR ideally will do is actually patch the security system, so there's no longer that opportunity for escape. And so that is one application I think for CRISPR. That doesn't actually even require you to change the sequence of the genes themselves, but this is something that is in clinical trials now.
Yep, that's right. Anybody else want to take that one on? CRISPR? If not, there's a couple of other really great questions I'd like to get to. One of them, Ali, maybe you could take this one. I love this. Is all the research at Kennedy Krieger about rare disease?
No, not entirely. So we have a wide array of research fields that we investigate, ranging from basic research to clinical trials. it's pretty wide scope. It's while the main focus is around developmental disabilities in children, that ranges from genetic diseases like these rare diseases we're talking about affecting the brain, but there's also neonatal brain injury, for example. We have a great emphasis and a great investigator group that's studying what happens to the brain after neonatal brain injuries or in preterm babies or babies that have lack of oxygenation at birth. We have investigators who are studying spinal cord conditions in children and in adults. We have, for example, an investigator who's focused on infections that result in paralysis of the spinal cord. We have investigations in mental health disorders that are affecting children. We have several clinical trials that are focusing on psychiatric illnesses in children, particularly those with autism who have also like depression, anxiety disorders, obsessive compulsive disorders, and so on. So it's really a wide array of research that we do. And rare disease is a good fraction of it. I wouldn't be able to tell you what percentage, but not all of it, no.
So a question came in what way, if any, will the emergence of AI assist in the diagnosis or treatment of rare genetic disorders? And I think any one of you can jump on that very good question. Ali mentioned it when he was talking about the N of 1 type of studies, but anybody want to take that one on?
I mean, I can talk a little more
Sure.
about it, just at risk of talking too much. But yeah, machine learning applications are happening at multiple levels. So it ranges from utilizing machine learning in the diagnostic field that's been going on for a long time. In fact, since pretty much genetic testing became commercially available, there has been computer applications that screen the genome, analyze them, align the genomes, and so on. So there's machine learning has been a great component of genetic analysis for many, many years. It's becoming more sophisticated in terms of trying to predict that changes that we see on the genome, how it would affect the protein, the result of that, sort of trying to understand what is the result of that change that we're seeing and trying to come up with prediction tools there. On the drug discovery side, we are now using machine learning tools to try to identify which drugs that are already on the market could potentially be fitting, sort of if we had a key and a lock system, and we say the disease is the lock, and what key could fit in there. That's something that you can do on a computational level. We use machine learning tools also now on identifying biomarkers, like Connie mentioned earlier. You may have two kids with two girls with Rett syndrome mutations or changes in their genes in the Rett syndrome gene, but they may present very differently. And we're using machine learning tools to look at blood markers and see if there are some signatures there that could allow us to understand who's going to develop the disease more severely. And so the traditional statistical approaches don't work there usually because there's too much data there, because we can get a lot of data right now. And so using machine learning often helps in solving there. But it's rapidly evolving. It's changing day by day. It's a little different than the ChatGPT stuff that you see online. So this is more different type of machine learning tools that are being applied in the labs often.
Connie, a question for you. It's related to rare disease, but it's really a general question about how can a family make sure that the gains made at Kennedy Krieger will hold while the child's at home?
Excellent question. One of the things that I often encourage parents to do is when they work with their therapists to ensure that they are learning what it is that they need to be doing to sustain or maintain those skills. If you think about how we all learn, we learn through practice. And as little kids, we're essentially repeating the activity. And so it becomes really critical that the therapies that the patient engages with at Kennedy aligns with the therapies that are being conducted in the school and also in the home. And if we keep repeating the same patterns, then the connections in our brain called the synapses become strengthened. And so it's very, we have seen it over and over. Early intervention has resulted in improved functioning for our patients.
So I think we have time for one more question, but it's a big one, and it has to do with how the research in this space is funded. Can you speak to the role that the NIH plays in funding work for rare disease, especially around treatment, development of treatments, and the role that pharmaceutical companies play? And it's a big question. Erica, you want to start out with that one?
Sure. The NIH is really a critical partner in support of research. Someone earlier talked about different kinds of partnerships, and in a sense, your funders, to a degree, are partners. And the NIH sparks and sponsors a lot of the work that happens in academic centers around the country. When we think about drug development in particular, there are a whole spectrum and hosts of various partners, and each kind of partner may have a different balance or portfolio of funding, but they also have a different balance or portfolio of skills. And the academic environment is a place where we are really well suited to understand mechanism of disease. Why and how does a disease happen? What is the pathologic biology of that disease? That is where expertise in academic centers lie. There are many academic centers that are also then discovering, just like Dr. Fatimi was just talking about, that lock and key idea. What are the kinds of targets that might unlock the idea of a new drug? And even to do some of that early work around developing those drugs. And that body of work tends to be funded by the NIH and similar kinds of funding partners. Later on, there are groups that are really, really great at getting drugs ready for testing in people, and the pharmaceutical industry is where those large-scale trials, someone asked about how do we bring together enough people to actually learn in a trial. The pharmaceutical industry is really great at the science of clinical trials and clinical trial design and answering those questions. The NIH plays a role there too, and many of us are involved in federally funded clinical trials that might be about developing drugs that come to market, but often they are about testing and answering questions that any one of us will really need to know, answer, use day to day in the clinic. So if there's a drug that's already on the market, I might have a really specific question about how I can use that. The NIH is likely going to be a sponsor and supporter for that kind of work. But again, we all have these roles to play, and there are different kinds of funds that support that. At Kennedy Krieger, we receive funds and support from the NIH, from other federal agencies, from foundations and nonprofits, as well as the pharmaceutical industry and other biotech.
That's great. And I'm afraid that's all the time we have tonight, but what a great conversation, outstanding discussion we've had this evening. I want to thank our expert panelists for joining us tonight, for sharing their insights and expertise. I especially want to thank our audience for your extremely thoughtful questions. It's always the best part of the evening is hearing what you're asking. So we hope that you found this webinar interesting and informative and helpful. And likewise, we hope that you, as a supporter of Kennedy Krieger, recognize and are proud of the major contributions we're making to improve the lives of people living with rare diseases. Now, in the next few days, you're going to receive a follow-up email with more information about the work that happens here at Kennedy Krieger. In addition, you can visit our website, kennedykrieger.org, where you'll find ways to support us, details about other upcoming events, links to our monthly podcast, Your Child's Brain, which is produced by Kennedy Krieger with assistance from WYPR. And finally, we hope that you sign up for future webinars in our Exploring the Brain series. So we're very grateful that you were here with us tonight, and thank you. Have a wonderful evening.